Hello, everyone. My name is Dr. Abhinav Choudhury. I am an environmental research engineer at the Freshwater Institute in Shepherdstown, West Virginia. I have worked on waste to energy and bioenergy technologies, specifically in the area of anaerobic digestion and life cycle impact assessment, which is used to evaluate the environmental impacts of these bioenergy technologies. Today, I'll be talking about the application of anaerobic digestion for converting aquaculture waste into energy. We'll start off with a little introduction on waste generation in aquaculture systems and how this waste can be used as a valuable resource. Then we'll focus on RAS waste and how we can utilize it for anaerobic digestion. Following that, we'll get into some of the basics of anaerobic digestion, and I'll talk about how we can use lab experiments to test the degradability of different waste materials. Then I'll talk a little bit about how we can improve the digestion process through the control of a byproduct known as hydrogen sulfide and through something known as co-digestion. Finally, I'll talk about how we can design a closed loop system for RAS facilities by utilizing the waste to its maximum potential. We all know that aquaculture is the hottest and fastest growing food production sector, which is also why many of us are in this industry in the first place. Growing demand for seafood has led to the intensification of aquaculture production facilities, leading to generation of a concentrated waste stream. This waste stream is a mix of fecal matter and wasted feed and can even include small amounts of biofloc and mortalities. This concoction results in a wastewater stream that is rich in organic matter and dissolved nutrients. Now, the discharge of this waste streams directly into the environment is not possible due to environmental regulations, as it can lead to the pollution of water bodies, eutrophication, and biosecurity concerns. As a result, production facilities have to bear additional disposal and treatment costs for the waste that is generated. As the number of production facilities keep growing, so does the need for an effective and a sustainable waste treatment system. In many of the newly designed fish production facilities, like partial reuse systems and recirculating aquaculture systems or RAS, the water is being reused, but the waste or the sludge isn't. And this waste is a valuable resource due to its high organic matter content, nitrogen and phosphorus content, along with other trace minerals like iron, zinc, etc. Now, the organic matter contains carbon, which can be converted into energy via thermal, chemical, or biochemical pathways. For example, in Baltimore, municipal solid waste, which contains a lot of carbon, is burned to produce energy in the form of electricity. In addition, nutrients like nitrogen and phosphorus, along with other trace minerals like iron and zinc, can be used for plant growth. For example, in dairy farming, the manure is used as a fertilizer to grow crops instead of being disposed of as a waste. Aquaculture waste also meets the criteria for being utilized as a resource for energy recovery and fertilizer production and use for, let's say, in a hydroponic system. Let's take RAS as an example of a fish production system because it generates a concentrated waste stream. RAS production facilities are gaining a lot of steam because of high biological security, continuous operation throughout the year, and enhanced control of the culture environment, along with minimal water usage. RAS also allows for efficient solid waste capture and collection, but as I previously mentioned, it results in the production of a concentrated waste stream, which can lead to extra disposal costs for production facilities or farmers. Aquaculture farmers may also have to bear additional costs for disposal of mortalities and offal, which can sometimes be more than 40% of the fish weight, and other byproducts during fish harvest. Studies have suggested that for salmonid farms, mortalities alone can be up to 9.5% of the total production. So for example, for a 1,000-ton farm, that's close to 100 tons that's not being utilized. And somehow, all of this waste that is generated from these facilities have to be treated or managed. So It leads to the question, what if we could use this waste to generate energy and a fertilizer stream? The energy could be used for the fish production systems, and the fertilizer stream could be used to grow plants in a hydroponic system, perhaps leading to another potential revenue stream. And that's where anaerobic digestion comes in. So what is anaerobic digestion? Well, it's a biological process occurring in the absence of oxygen, where microorganisms degrade organic matter in the waste, producing a mixture of methane, carbon dioxide, and other trace gases like hydrogen sulfide, which can be a problem. We'll get to that later. This combination of gases is also known as biogas. The energy content of biogas, or rather methane, which is also the main component of natural gas, can be used to run boilers or generators for heat and electricity production. Anaerobic digestion process has 4 main steps, as you can see in the slide, and methane is produced in the final step. I'm not going to get into the details of the 4 steps in the presentation, but please email me if you're interested in knowing more about the biochemistry behind the process. Anaerobic digestion systems are currently used on dairy farms, swine farms, and at wastewater treatment plants to produce energy to partially or sometimes even completely meet the energy demands of the facilities. Usually, at these facilities, the manure, in the case of dairy or swine farms, or the wastewater sludge in wastewater treatment plants is fed into the digester to produce energy and a fertilizer stream. In a similar manner, anaerobic digestion could also be used to convert aquaculture waste into renewable energy with mineralization of nitrogen phosphorus. Mineralization basically means the process will convert organic and phosphorus into plant available dissolved forms. Anaerobic digestion also reduces the pathogen content and solid concentration of the waste, sometimes up to 80% or more. In addition, it may also be possible to recover anywhere from 2 to 18% of the energy requirements for RAS systems from this process, which can also lead to additional savings for a production facility. So how do we test if aquaculture waste is a feasible feedstock for anaerobic digestion? Well, we can do this by using a biochemical methane potential test. In this test, we add the carbon containing substrate, which in this case would be the aquaculture waste. The inoculum source is usually the effluent that comes out of the existing digester. We mix both of these up, then we purge the headspace of the reactor bottles with nitrogen to create anaerobic conditions. We incubate the bottles for 30 to 45 days at 35 degrees Celsius. The experimental time usually depends on the biogas production rate. And finally, we measure the volume of biogas that's produced and quantify the methane concentrations. We can also quantify the carbon dioxide concentrations and the hydrogen sulfide concentrations, if necessary, using an analytical technique known as gas chromatography. Through this methane potential test, we can also estimate the biogas and methane production of a mixture of wastes. For example, the waste that is generated, let's say the offal that is generated on a fish during fish harvest, we can add that fish offal into the mix. Fish offal also contains carbon, so in theory, it should help enhance the methane production, which leads us into what we call co-digestion. Co-digestion of organic waste is basically the digestion of waste organic matter, such as agricultural waste or food waste, along with dairy manure or wastewater sludge. Basically, it means the mixture of two different kinds of waste, two or more different kinds of waste, instead of just digesting one kind of waste. In the case of aquaculture systems, it could be the addition of fish offal into the aquaculture waste stream. The advantage of co-digestion is that it can help increase biogas production dramatically in some cases and result in a higher return on investment for aquaculture practitioners. So while I was doing my PhD, I worked on co-digestion on gummy vitamins with dairy manure. I was working on a dairy farm in Maryland, Pennsylvania, and New York, and on one of those farm visits, we saw that a farmer was interested in using a gummy vitamin waste as a co-digestion feedstock for their dairy manure digester. And they wanted to know whether they could add this material to their digester to further increase the biogas yield. We were intrigued, obviously, and so we took this material and conducted a biochemical methane potential test to quantify its methane potential. We mixed the gummy waste with dairy manure, and the results showed that the addition of gummy waste resulted in a 5-fold increase in methane yield when we compared it to digesting dairy manure by itself. Similar results have also been seen by other researchers. For example, my PhD advisor observed a 26-fold increase in methane yield when they mixed chicken processing waste with dairy manure. So there is potential for increasing the methane production when we mix other kinds of waste with aquaculture waste. In this graph, you can see that a lot of the co-digestion of different feedstocks can produce a lot more methane than the digestion of just dairy or swine manure. In some cases, like for food grease, it can result in a multifold increase in the methane yield per ton of the added material. Most previous research on this topic has been focused on methane potential of the different materials that can be used for co-digestion. Co-digestion can help balance factors that control anaerobic digestion, like ammonia concentrations, carbon-nitrogen ratios, pH, volatile solids content, and volatile fatty acid content. Let's take aquaculture waste, for example. For example, aquaculture waste solids tend to have a CN ratio that is below the optimal range of anaerobic digestion process, and it could have a detrimental effect on methane production, which in turn can reduce the energy content of the biogas. In such case, co-digestion of other organic substrates with a high CN ratio with the aquaculture waste may be an effective way to enhance the methane production and improve energy generation by balancing the CN ratio. Co-digestion can also help with some other factors, such as hydrogen sulfide production, which is usually a problem in anaerobic digestion systems. So why is hydrogen sulfide a problem? Well, it's because hydrogen sulfide is corrosive, and it can damage the digestion system and the energy generation equipments. The waste material that is fed into a digester usually has sulfur-containing compounds. These compounds are broken down by a group of microorganisms called sulfate-reducing bacteria, or SRBs, which results in the production of hydrogen sulfide, or H2S. In the slide, you can see the corrosion of the generator and the pipeline due to hydrogen sulfide. Sulfate-reducing bacteria, or SRBs, can also inhibit and reduce methane production because they compete with methane-producing microorganisms for carbon in the feedstock. So if there is a high hydrogen sulfide production from a digester, there's a high likelihood that the methane is not being produced optimally. Hydrogen sulfide production can be controlled through physical, chemical, or biological methods, but like you'd expect, it would result in increased capital and operating expenditures for the farmer. I've worked on novel low-cost methods for hydrogen sulfide control in anaerobic digestion systems, so if anyone is interested in knowing more about it, please contact me and I'll be happy to share some of the information. So we talked about anaerobic digestion and how it can be used to produce methane and energy from the carbon content in the aquaculture waste. We talked about co-digestion of fish offal that could be used to enhance the methane yield. We briefly touched on how anaerobic digestion can also be used for fertilizer production through the mineralization or breakdown of the organically bound nitrogen and phosphorus in the waste, and how we can utilize the dissolved nutrients in a hydroponic system. When you combine all of this together, we can design a closed loop system for managing and treating the aquaculture waste from a RAS facility. And this is what I expect the final system to look like, as you can see on the slide, where the waste from the RAS system goes into the digester. The energy that is produced in the form of heat and electricity can be used to maintain the digester temperature or be used to recover the energy needs for the RAS systems. Then the effluent from the digester with the mineralized nitrogen and phosphorus and other trace minerals could go into a solid separator, where the liquid and the remaining solids in the effluent could be divided into two different waste streams. The solid fractions could be composted and used as a solid fertilizer or transported off-site, but the liquid fraction with all the dissolved nutrients can be used as a fertilizer for hydroponic system. Once the plants in the hydroponic system absorb the nutrients, it could potentially be fed back into the RAS system after removing the residual ammonia in the nitrification bioreactor. Finally, the hydroponic system would be producing vegetables that can be sold at the market for an additional revenue source. However, the vegetable waste, like the stalks, the leaves, could be fed back into the digester as a co-digestion material as well, thus closing the entire loop for the system to a much higher extent. And that is not all. It could even be possible to add the offal and mortalities into the digester to enhance the methane yield. Through the system, the amount of waste that requires disposal off-site could be significantly reduced. At the Conservation Fund Freshwater Institute, our goal is to conduct the research to make this idea feasible. And with that, I will end my presentation. Thank you. My email address is on the slide, and I would like to thank USDA for funding the ongoing research at the anaerobic digestion at the Conservation Fund Freshwater Institute.